Dov's topic for us today will be applications of NGS for molecular pathology, diagnosis of tumors. Thank you, Dov.
The field of molecular pathology developed or started 10 to 15 years ago, and with the introduction of NGS platforms that can be applied in-house, this has revolutionized the way we make diagnosis in cancer. And the purpose of the presentation today is to tell you a little bit about how we use NGS technology in molecular pathology diagnosis. And I will talk about NGS use for isolated point mutation and for comprehensive comprehensive panels. But first, since I think that many of the audience here are germline geneticists, I would like to speak a little bit about the difference between somatic genetics and germline genetics. In my view, cancer is a genetic disease because cancer is caused by mutations in the DNA. The consequence of these mutations is alteration in cancer hallmarks. This could be activating mutations in oncogenes, loss of function mutations in tumor suppressor gene or genes related to apoptosis, and also evasion of the immune system. And when we talk about germline mutation, these mutations are mostly inherited from one of the parents or both of them, and these mutations are present in the fertilized egg. The meaning is that these mutations will be present in all the cells of the body. On the other hand, when we talk about somatic mutations in cancer, these mutations are mutations that are acquired throughout life and are selected and are present only in the tumor cells. So this has some important clinical and practical implications. For example, to detect germline mutation, we can take a culture fibroblast, we can take swabs from the patient, we can take blood samples, and we'll get the same mutation mostly in all the samples. On the other hand, for somatic mutation, we must have the tumor tissue to make the diagnosis, and even more so in the same patient, different mutation, for example, a primary tumor and different metastases can have different type of mutations. So this is the spatial variation that we can see with somatic mutations in cancer. Another difference is the temporal variation. When we are talking about somatic mutation, they change with time, either because of selection within the tumor or following treatment. On the other hand, germline mutations are relatively stable throughout life. So how does this affect the way we make diagnosis in cancer? For example, one important thing is tumor selection. We perform the analysis on a tumor sample, and we need to select a sample that contains a large enough amount of tumor cell. The tissue samples from the patients are not cell culture containing 100% tumor cells. It contains many other cells. So it's very important from what type of tissue we extract DNA and perform the analysis. For example, if we look on the sample to the left, in this sample, there is, like 80% tumor cell content, and you can see the Sanger sequencing. We can see a very clear mutation here. On the other hand, in this sample, there is mainly fibrosis, reaction of the stroma, and only isolated tumor glands. In this sample, we were unable to detect the mutation using Sanger sequencing, even though it was identified using a more sensitive method. So tumor selection or selecting the right tumor area is important for making an accurate diagnosis. Okay, but we are here to talk about NGS. And why would we want to use NGS for isolated mutations? One reason, one advantage of the NGS is that it can tell us the mutant allele frequency. Okay, classically we think of tumors as a clonal disease. That means that the tumor cell either contains a specific mutation or does not contain a specific mutation, okay, black and white. But in life, we know that there are more shades to the reality. So if we look at it schematically, the old understanding is that tumor can be either wild type, that means that none of the tumor cells carry a specific mutation, or it can be carrying a mutation, that means that all the tumor cells carry the mutation. But in real life, we see other conditions as well. So in some tumors, we can see intratumor heterogeneity. That means that some of the tumor carry the mutation, and some of the tumor cells carry the mutation, and some of them don't carry the mutation. And we can also see mutant allele-specific amplification. So using next-generation sequencing, we can determine the mutant allele fraction in the sample we analyzed. And using this information, it is possible to determine whether a specific mutation is clonal, present in all the tumor cells, or subclonal, present only in the fraction of tumor cells. Of course, it's important to remember, as I said earlier, that the tissue sample that we use for diagnosis does not contain only tumor cells. For example, here the brown cells are, this is papillary thyroid carcinoma, and the brown nuclei are from tumor cells, and these are lymphocytes that we do not expect them to carry the driver mutation. So we need to analyze the tumor cell fraction in the sample that we are dealing, and based on this, we can give the correct interpretation for the mutant allele fraction in the tumor. In other words, if the mutant allele fraction is equal to the tumor cell content, that means that for this specific mutation, the tumor is clonal. All the cells carry this mutation. On the other hand, if we see a mutant allele fraction that is considerably lower than the tumor cell content, than what we would have expected, that means that the tumor is subclonal. This mutation is subclonal in the tumor, or in other words, called intratumor heterogeneity. We applied this approach to several different tumors, and we were able to show that for the major drivers in these tumors, it was BRAF mutation in thyroid carcinoma and KRAS mutation in pancreatic carcinoma. We were able to show that for this mutation, subclonality was associated with earlier disease in thyroid carcinoma, smaller tumors and tumors that do not invade to the surrounding, and in pancreatic carcinoma, it was associated with prolonged survival. So that's one application of NGS for a single isolated mutation. Another interesting application for isolated mutation is the application in liquid biopsy. A good example to explain this concept is lung carcinoma. About 20% of the patient with the most common type of lung cancer, it's lung adenocarcinoma, carry a mutation in the gene EGFR. The consequence of this mutation is constant activation of the proliferative signal. Unfortunately, there are several drugs that have been developed that target the mutated protein and are very effective in this patient. So we want to make this diagnosis, and here you can see a summary of a number of very large clinical trials, and as you can see here, the green are the EGFR specific therapy, the inhibitors. They are called tyrosine kinase inhibitors, and the gray color is chemotherapy. And as you can see, the biological therapy in patients carrying EGFR mutation was more effective. But we can see another thing here, and even under the targeted therapy, between after something like 9 to 13 months, the disease progressed. Okay, and why did the disease progress? This is because of resistance mutation. Okay, the tumors change with time. There is resistance mutation, and the most common mutation is very easy to detect. It's T790M mutation in EGFR. So we have a patient that was treated for lung cancer very effectively, but about 1 year after the beginning of treatment, the disease, the treatment that was effective stopped being effective, and the disease is progressing, and we want to make a diagnosis whether the resistance mutation is T790M. This is important because there is a new drug that targets T790M. Okay, so we want to make this diagnosis. So we talked already, diagnosis is made on tissue. So we need to take a tissue. So there are different ways to take a tissue. All of them are invasive. Okay, invasive and contain some risk. And here comes to our help the liquid biopsy. So it appears that tumor cell content are released into the circulation. Okay, this can be exosomes from the tumor, it can be DNA, nuclear DNA from cells that died, and it can be whole cells. Okay, they can be detected in the blood, and if we have a test that is sensitive enough that can detect mutations in the circulation, we can perform the analysis without causing the invasive and risky test for the patient. Another advantage of liquid biopsy over regular biopsy is that biopsy represents only the small sample that we took from the patient. Okay, if a patient has a primary tumor and we took the sample from the primary tumor, we'll see only the mutations of the primary tumor. And if he has a metastasis and we took the metastasis, we will see only the mutation in the metastasis. But in the blood, we have a representation of all the mutations that are present in the body. So this is another advantage. So different tumors, the most common type, the most widely used type of liquid biopsy is circulating tumor DNA. And ctDNA can be detected in different types of tumors. Different tumors have different levels of circulating tumor DNA. For example, colon cancer and bladder cancer have high levels of circulating tumor DNA as opposed to brain tumors, for example. So the type of tumor affects our ability to detect this mutation in the circulation, and also the stage of the tumor. Affected, earlier stage, smaller tumors are associated with less circulating tumor DNA. But the big challenge in detecting mutations in liquid biopsy is that there is a very low fraction of the mutant DNA in the circulation. In the plasma, the amount of circulating tumor DNA is very low. And here NGS can come to our help because we can use ultra-deep sequencing, and using ultra-deep sequencing we can detect very low frequency mutation, including mutations that are less than 1% mutant allele fraction. So this is an application that we can use. We actually did recently a study. We followed patients with pancreatic adenocarcinoma that had KRAS mutation, and we followed KRAS mutation level, allele frequency in their blood over time. This was done in collaboration with Dr. Uti Peretz from Oncology Rambam and Dr. Orly Greenberg, who is a resident in our department. And as you can see in this graph, that there was a good correlation between ctDNA levels and CA99, which is the protein marker for pancreatic cancer. Another thing that we can see that following an increase in the levels of KRAS was associated with very short survival. Okay, following the increased patient, the asterisk is the time of death. So following an increase, the patient rapid decrease. The patient died shortly after. Okay, and this goes in line with the concept that increase in circulating DNA is associated with disease progression, whereas decrease in the levels of circulating DNA is linked to good treatment response. We actually also measured the slope between two following measurements, and the changes in ctDNA level in two following measurements. And as you can see, low levels or downward slope was associated with longer survival time as opposed to upward slope that were of course associated with very short survival time. Okay, so this is the application of NGS for point mutation, isolated point mutation. But with no doubt, the biggest advantage of next generation sequencing is for more comprehensive panels. And in the last years, there was a diagnostic trend from single point mutations to gene panels, I don't know, a few dozens of genes that are of interest, and now we go toward the larger panels of hundreds of genes. And the most extensive, the most comprehensive panel that we are currently using is linked to the Nobel Prize in Medicine 2019 that was announced a few days ago. The prize went to James Ellinson and Tasuku Honjo, and they got a prize for discovering cancer therapy by inhibiting negative regulation of the immune system. So what is the basic concept? The mutations in the tumor cause change in protein. This changed protein is perceived by the immune system as a non-self, and this causes attack of the immune system on tumor cells. What tumor cells do? They cause regulatory pathways that block this attack by the immune system, and the therapy that was developed inhibits this negative regulation, and therefore the immune system can attack the tumor. So we know that different tumors have different amount of mutation. It's called different tumor mutation burden. For example, the tumors with highest tumor mutation burdens are melanoma and lung cancer. And conceptually, tumors that carry lots of mutation will have lots of changes in their proteins, and therefore should respond better to therapies that activate the immune systems against the tumor. And this actually is a correct prediction, as you can see, with the exception of brain tumors. Okay, in all the other types of tumors that were evaluated, higher tumor mutation burden, more mutations within the tumor was associated with better response to immunotherapy, and with the increased use of these drugs in the clinical practice in the treatment of cancer, I think that there will be an increased utilization of this new biomarker tumor mutation burden. Okay, and finally, I would like to speak about a more challenging aspect of comprehensive cancer diagnostic, and is the detection of gene rearrangements or translocations. And just for example, soft tissue sarcomas are malignant tumors of the soft tissue and the bones. And interestingly, many of these tumors carry a driver translocation. And these translocations are used to make the accurate diagnosis and also affect treatment decision. Just one example, DFSP or dermatofibrosarcoma protuberans is a subcutaneous tumor. It is caused by a translocation involving the collagen gene and the oncogene growth factor PDGFB. The consequence of this translocation is that we have lots of the growth factor PDGFB, and this induces a signal, a proliferation signal in the fibroblasts, and that's the basis for this disease. So we want to make diagnosis of these translocations. Classically, we use FISH, fluorescent in-situ hybridization, to make the diagnosis. The commonly used approach is break-apart probes. We use two fluorescent dyes that are present on both sides of the predicted breakpoint for the translocation, and positive result will be, if there is a real break, we'll see break-apart signals. That means that the two signals came apart from each other. There is a problem or two problems with this approach. First of all, we need multiple probes because there are multiple diagnoses. And the other problem is that sometimes same gene with a different partner means a different diagnosis and a different treatment. So just knowing that there is a break apart involving a specific gene is not enough. And so we need a comprehensive panel. I don't know if you can see, but you can get the impression that this is a very large list of translocation that we are interested in to make the diagnosis. So this is a challenge. There are two ways to make the libraries for this diagnosis. It can be PCR based or a hybrid capture. I won't tell you about the differences, technical differences between PCR enrichment and a hybrid capture, but on the practical level, PCR or amplicon-based approach is associated with shorter turnaround time, lower cost, but the disadvantage is for unique partners. Okay, we need primers from both partners of interest to make the diagnosis, and this way we cannot identify unique or novel translocation partners. But in this respect, there is a new technology that we are using. It's called Archer Fusion Plex. This is a PCR amplicon-based approach that can identify also unique partner, and how this is being done. Okay, after ligation of the adapters and barcodes, there is a PCR reaction, but the PCR reaction uses only one gene-specific primer only from one direction, and the reverse primer sits on the adapter. So this way we get a PCR product, but it's unique only for the first gene partner. And in the sequencing reaction, we can also identify unique partners. And this way we have both the advantage of PCR amplification and the ability to identify new targets. So that's something of interest. So if I want to conclude, so this is an exciting time for pathologists, and NGS already has a very central and important role in the clinical diagnosis of cancer, and our mission is that the young pathologists, the young residents who will need to use these approaches in the routine clinical practice in the future, we need to teach them and to give them a better education in genetics. Okay, thank you very much.
If there are any questions?
How do we validate the results after the Archer fusion plex after the NGS?
We don't perform additional validation. There are criteria in the Archer. We need to identify unique. There are different start sites, and we identify different products because the reverse primer is the consequence of the ligation. So if we get enough different products, it's called reliable. Okay, because it's not just PCR. It's not one product that can give all the results. We see sequencing of the gene of interest and the partner, and we can see different such sequences. Okay, because the reverse primer is on different areas. So there are criteria to call a translocation real, and you don't perform additional validation. Yes.
Also regarding the translocation assay, so most of the events that we're going to sequence are going to be without translocation.
Yes.
One way to overcome it is, because we are also using for the genome editing field, is to use a restriction enzyme that get rid of all the events that are not translocated, and then you can really build on the deep sequencing without using it to sequence most of the wild type events. This is something very easy to add to the procedure and can add a lot.
In our that's a very good idea. In our experience, usually in sarcomas, in this type of tumor, the tumor cell content is very high. So the events are not that rare. So with a reasonable coverage, we identify. I'm not sure about the significant added value for getting rid of the wild types. It's
beautiful. Just going back to the previous question, I think what just for me to hear myself saying it, what you're saying is the location of the sonication break of the fused gene is
exactly
variable, and so
exactly.
you see the same gene product starting from multiple templates, and that's why you
and that exactly. Thank you.
But
Thank you for the
question.
thank you for the explanation. And so I see different supporting, different types of supporting, different families of supporting reads, and this increases my confidence in the diagnosis. Thank you.
My question is on the work where you are tracking the KRAS and the CA19. Did anyone run power calculation on this? Do you know how many patients you need to track to make that statement? If there's an increase, they die younger, or?
This was relatively sporadic. It's very difficult to collect a large cohort of patients that are followed for a long period. We didn't perform power analysis to draw conclusion, but without power analysis, this is very.
You're not really tracking multiple variables here. There really is. It shouldn't be so hard to get to power.
Yeah.
You're not tracking hundreds of thousands.
We didn't do it, but yeah, it shouldn't be difficult. Yes, please.
When addressing genome rearrangement, is it possible to use a method that sort of mimics RNA sequencing that handles exon-exon junctions? When you break apart all your sequencing components, and you need to align them to the genome, and you have some parts that are not in their proper location.
Okay, you're talking about the bioinformatic analysis.
Yeah.
Actually, the analysis, this process is performed on RNA. Okay, it's very in the DNA extracted. I didn't mention it, but we extract DNA from formalin-fixed paraffin-embedded tissue, and this DNA is fragmented, so it would be impossible to get large enough products to identify this translocation at the DNA level. So we performed the analysis on the RNA. Actually, I don't know much about the bioinformatics. We get it with a software tool that we can apply for making the diagnosis. Okay, so yes.
How widely is the NGS used in diagnostic?
Very widely. We perform an NGS reaction once a week for something like 20 patients. So this is something very routine in clinical practice. Actually, today, every patient with melanoma, every patient with colon cancer, every patient with lung cancer, all of them get, and it's covered by the health insurance. every patient gets NGS analysis for the relevant gene. So this is very widely, it's daily practice. Okay, thank you very much.